In all 7 traits that Mendel studied in pea plants, there was one gene that affected one trait, and each of those genes had only 2 alleles. This is a good way to learn about genetics, and Mendel had luck in that he picked 7 traits that followed the rules that he would establish. But the fact is that in most traits, there are multiple alleles or multiple genes that determine those traits. And so the Mendelian rules, and otherwise the rules established by Gregor Mendel, the Mendelian rules for genetics don't always hold true in real life. They form a basis for what we understand, but we now understand much more, and we can describe several other patterns of inheritance, which we are going to learn about today. When Mendel examined the characteristics of pea plants, he was fortunate in that the particular characteristics were each controlled by a single gene, each of which had 2 alleles. Think about all the different shades of hair color that humans have. Do you think hair color is controlled by just one gene? It would be hard to imagine that it's controlled by one gene that is only dominant or recessive, because that really only allows for 2 different variations. The reality is that something like hair color is controlled by many different genes. Today we will describe some of the exceptions to Mendel's principles and explain the environment's role in the way genes determine traits. There's a type of plant called a 4-o'clock plant. The 4-o'clock plant has red flowers, white flowers, or pink flowers. It uses or it follows a different pattern than Mendel saw with pea plants. 4-o'clock plants, their flowers have genes for flower color, that don't follow the strict dominance pattern that Mendel saw in different characteristics of pea plants. In 4-o'clock plants, the alleles for red and white flowers show incomplete dominance. Heterozygous plants have pink flowers, a mix of red and white coloring. You have a dominant allele, which is the red allele, and a recessive allele, which is the white allele, but the red allele is not completely dominant over the white allele. It's sort of a partial dominance. And so when you mix the red and the white, and you get the heterozygous plants, you get some red and some white mixed together, and you get the pink flower. So the parent plants in this cross, in this genetic cross that we see in the example, are both homozygous, and we see the offspring are heterozygous, and those heterozygous offspring get some of the red color and some of the white color mixed together to make a pink color. So could plants with pink flowers produce any offspring with red flowers? If you took these offspring and did a dihybrid cross, think about what the Punnett square would look like. You would get, in the top left part of the Punnett square, you would get a red flower with two dominant alleles. In the bottom right corner, you would get a white flower with 2 recessive alleles, and in the other 2 boxes, you would get more heterozygous plants, which would turn out to be pink. So your ratio would be 1 red to 2 pink to 1 white. In codominance, you also don't have complete dominance of one allele over the other. But it's different than in complete dominance. In codominance, the phenotypes produced by both alleles are clearly expressed. In some varieties of chicken, the alleles for black feathers is codominant with the allele for white feathers. So heterozygous chickens have a color described as erminette. The phenotype of the chickens, they come out speckled with both black and white feathers. The black and white alleles are codominant. So how is incomplete dominance different from codominance? In incomplete dominance, the traits are blended. You get something that is somewhere between the two alleles. In codominance, you have both traits distinctly expressed. You have something like this chicken, which has black and white feathers. You may be familiar with the concept of blood types. When you go to the doctor, if anytime you need blood, they have to provide you with blood from a donor that has the same blood type or a compatible blood type. The terms you may hear are A positive, B positive. So you have A, B, and O blood types, and you also have an AB blood type. And then for each of those four variations, there are positive and negative. Positive and negative is what we call the RH type. This happens because there are more than two alleles for the blood type gene. You have A, B, and O, three different alleles. This is very common in a population. Any given individual in this population, though, only has two alleles. You have two alleles that determine your blood type, and then two separate alleles that determine your RH, positive or negative. In the case of human blood types, some alleles also show codominance. The A and B alleles are codominant with each other. A and B are each dominant over O. So O is truly recessive to A or B, but A and B are codominant. The RH factor is inherited independently of the blood type alleles. RH plus is dominant over or RH positive, we call it, is dominant over RH negative. So this is why you have two separate parts of the name for your blood type. You have the ABO or AB blood type. Remember, A and B are codominant. So if you have an A gene or an A allele and a B allele, you are AB. If you have two A alleles or an AO allele, then you are type A. If you have two B alleles or a BO or a B allele and an O allele, then you're type B. And if you have two type O alleles, then your blood type is type O. If you have one A allele, one B allele again, they are codominant. You end up with blood type AB. So you might say that I am A positive. That's a very common blood type. A positive. That means that you have either AA or AO, and then you also have the positive RH type, because RH is dominant over, RH positive is dominant over RH negative. Look at this example of rabbits, rabbit coat color. So these rabbits have multiple alleles. The alleles occur in order from the most dominant to least dominant. Capital C is full color. Capital C with a little CH is chinchilla color. Then we have just the CH, which is Himalayan color. See, it's a C with a little H. That's Himalayan, so that's recessive. And then what's recessive to all of the other three alleles is albino. Albino rabbits have no color. If you made up genetic crosses for coat color in rabbits, you could see all the different variations of rabbits that could be produced. So we see here 4 examples that are produced, and this is one gene that determines coat color, but there are multiple alleles. There's not just one dominant and one recessive. There are 4 different alleles. Some are more dominant than others. Polygenic traits are produced by the interaction of several genes. Multiple alleles are different from polygenic traits because multiple alleles refers to different alleles for a single gene. What we saw with the rabbit coat colors, that was multiple alleles. There was one gene determining coat color, it had 4 different alleles. Polygenic traits refers to multiple genes influencing a single trait. Many students think that one gene is always responsible for one trait because that's what we learn first when we're learning about Mendelian genetics, but that's actually pretty rare. Mendel was very lucky that he picked 7 traits that followed those very strict rules. Most traits, such as hair and eye color in humans, are influenced by multiple genes. That's why we see such great variation in human hair color and human eye color. There's an insect called the western white butterfly, which is found throughout western North America. Over the years, some people noticed that western whites hatching in the summer had different color patterns on their wings than those hatching in the spring. Scientific studies showed the reason. Butterflies hatching in the shorter days of springtime had greater levels of pigment in their wings, making their markings appear darker than those hatching in the longer days of summer. In other words, the environment in which the butterflies develop influences the expression of their genes for wing coloration. The pictures here are not western white butterflies. They are, in fact, buckeye butterflies. And buckeye butterflies show a similar pattern. They're darker in the summer than they are in the autumn. The characteristics of any organism, whether plant, fruit fly, or human being, are not determined solely by the genes that the organism inherits. Genes provide a plan for development, but how that plan unfolds also depends on the environment. The phenotype of an organism is only partly determined by its genotype. Consider in the case of butterflies why being darker in the autumn than in the summer might be useful. In order to fly effectively, the body temperature of the butterfly must be between 28 and 40 degrees Celsius. That's between 84 and 104 degrees Fahrenheit. If it's too cold, they can't fly very well. If it's too hot, they overheat. Because the spring months are cooler in the west, greater pigmentation helps them reach the body temperature needed for flight. Similarly, in the hot summer months, less pigmentation enables the moths to avoid overheating. The term environment refers to internal factors too. For example, both men and women can have the genes for male pattern baldness, but baldness shows up far more often in men than in women because male hormones trigger the expression of the gene.